Hello, everybody, and thank you for joining us for our first Sampled Tech Talk. My name is Harris Chowdhury. I am the Product Marketing Manager here at Sampled. Today, I am joined by Dr. Jennifer Moore, who is Head of Cellular Services at Sampled. Jen, why don't you give us a little bit of background as to what you do at Sampled?
Sure. Hi. As Harris said, my name is Jen Moore, and I'm head of cellular services at Sampled. I've been at Sampled for about 12 years, and I started in their stem cell lab, which my scientific background is in pluripotent stem cells. Uh, I have recently become head of cellular services, which means not only do I oversee our stem cell team, but also our cell lab team. Our cell lab does PBMC processing, cell banking, and lots of other services related to cells, while our stem cell team does derivation of iPSCs from source cells, as well as banking, distribution, and quality control of iPSCs, and gene editing of iPSCs.
And that's actually what we're here to talk about today. So you mentioned gene editing, and there's a lot of buzz around CRISPR gene editing at the moment. Can you tell us a little bit about CRISPR gene editing, what it's used for, and some of the applications?
Sure. CRISPR gene editing is something that's fairly new. It's been around for maybe 5 or 6 years. And the great advantage of it is that the area that you can edit in a genome of a cell is very flexible. Some other types of editing that were popular in the past had very stringent structural and sequence requirements around the DNA, meaning that you had to have those requirements in place in order to do the editing. However, in the case of CRISPR, those requirements are very minimal, and that means that you have a lot more places in the genome where you can actually put your edit. People like to do lots of different kinds of editing. You can create a random break in the DNA and create a random insertion or deletion that disrupts a gene's function. You can change 1 or 2 or 3 nucleotides that can make a cell line go from a disease phenotype to a wild type phenotype, or opposite, a wild type phenotype to a disease phenotype. You can make reporter genes and other kinds of things to help you understand downstream assays. So basically, any way that you can think of to modify the DNA in the cell line, you can do through CRISPR gene editing.
I mean, that sounds very powerful as a technique. Is it something that's easy for people who have never approached CRISPR gene editing in the past to do themselves in their own labs, or is it a bit more tricky than they might expect?
I think it's trickier than they might expect. The reason is because in order to get an efficient edit, you really have to make sure you cut the DNA in the right spot, and you have to make sure that that cutting is sufficient to allow your edit to occur. And this means that sometimes you have to test multiple parameters, whether it's the guides that direct the cutting, whether it's the delivery into the cell, whether it's the concentrations of the different guides and CRISPR-Cas9 that you're delivering. All of those things make for a number of variables, and you have to change those variables in order to get an efficient edit. And so I think while people can learn how to do it, it takes a lot of resources and it takes a lot of time. And this generally takes away from the time that people that people would like to be using to pursue their main scientific question.
Okay, great. Can you tell us some of the advantages then, if someone wanted to do CRISPR gene editing and outsourced it to Sampled? Like, what are some of the advantages of that?
Sure. So other than the advantage of just time and resources, um, I think we have a lot of experience, and so we have already gone through a lot of permutations to understand what are the best guides, what are the best donors, what's the best delivery systems, how do you ensure that the cells are completely edited. So our experience basically makes the process quicker. And then in addition to that, we have a lot of quality control that's done standard on our cell lines that ensures that a cell line that someone gets from this process is high quality and functions the way they want it to. And then I think finally, we have an excellent reputation for really delivering high quality pluripotent stem cells that you can use in any of your downstream applications.
Fantastic. Thank you very much, Jen, for talking with us today, and we will see you on the next Sampled Tech Talk. Thank you.